Now we're going to move into our next session, which is innovative trial design. Our first speaker will be Darius Ebrahimi-Fakhari. Thank you.
Good morning, everyone. It's a great pleasure and honor to be here. Thank you for inviting me to speak. It's a great honor to be at this conference. So my name is Darius Ebrahimi-Fakhari, and I'm a pediatric neurologist. I direct the movement disorder program at Boston Children's Hospital, and my team has been working on a condition that is, in many ways, actually related to CMT, called hereditary spastic paraplegia. So I was invited to share a couple of lessons we've learned working on this rare disease. The title of my talk is Clinical Trial Design for Gene-Based Therapies in Ultra-Rare Diseases, Lessons for Hereditary Spastic Paraplegia. These are my disclosures. And before I go into this talk, I wanted to acknowledge that all of the work that you're going to see is made possible by many families and advocates who help us conduct clinical research studies. You'll see a lot of videos throughout the talk, and these were shared with permission. So in the next 15 minutes, I hope to introduce you to childhood onset hereditary spastic paraplegia, and I'll show you how this is in many ways a paradigm for rare disease research. I'll talk a little bit about preclinical development for gene-based therapies from concept, proof of concept to an IND. And then I'll share two examples of clinical trial designs for ultra-rare diseases, insights from early phase studies, and some thoughts on designing pivotal studies for ultra-rare diseases. And hopefully we'll have some time for questions during the panel session. So I'll start with an introduction to childhood onset hereditary spastic paraplegia. Like CMT, this is a large group of diseases that are clinically and molecularly heterogeneous. Monogenic diseases, they all have one thing in common. They present with progressive lower limb weakness and spasticity. You can appreciate this in this young patient of mine who has the most common form of hereditary spastic paraplegia, SPG4, and you see the spastic gait. This disease as a clinical entity was described over 100 years ago. You see the first description in the German and French literature in the late 19th century. But it wasn't until the 1990s with gene discovery that it was recognized that it's actually not one disease, but we now recognize over 90 different monogenic diseases that all present with these central clinical features. If you take the prevalence of all the HSP forms together, we're looking at about 2 to 5 in 100,000. So we're in the rare disease space. Although, as I'll show in the next couple of slides, this is probably an underestimation. And historically, this goes back to a clinical classification that precedes genetic or molecular classifications. We distinguish between pure forms, which means that an individual has weakness and spasticity, and complex forms, which present with additional central or peripheral nervous system manifestations. One of the, I think, reasons, not just our own experience, but one of the reasons scientifically why I think HSP is actually more common than we currently appreciate comes from related work in a much more common condition called cerebral palsy. So work over the last 5 years has established that if you use unbiased genetic testing, next generation sequencing-based testing, exome and genome sequencing, in cohorts of children and young adults with cerebral palsy, you actually find that 30% have a genetic diagnosis. And when you look more carefully, and I picked 3 studies in a meta-analysis to illustrate this, 25% of these 30% have hereditary spastic paraplegia. So that is good evidence that we're still missing a lot of cases, and the prevalence is actually probably much higher. We started working on this. There was very little known about childhood onset forms. So we started a natural history study about 5 years ago. At this point in time, we've met over 600 families from around the world. And similar to CMT, I would argue, we see a great clinical spectrum. We see a variety of clinical manifestations. I'm not going to comment on the videos, but just say that it illustrates how challenging natural history studies can be in this condition, because we're ranging from very young children with very severe symptoms to young adults with slowly progressing symptoms. So how do we capture that in a natural history study? We started this work at Boston Children's Hospital. We're very fortunate over the last 2 years to have had support from the Spastic Paraplegia Foundation to extend this work now to 11 sites across the US. This is a multi-center study, and I'm very pleased to say that we have now received funding through the RDCRN an NIH-funded study that will elevate this consortium to the next stage. What have we learned? From this effort, we've learned a great deal about autosomal dominant forms, recessive forms. We've learned what was just presented in CMT as well, that despite the advanced genetic testing that we have available today, about 30% of patients do not receive a molecular diagnosis. So we're closing this gap by using more advanced genetic testing. And finally, as a clinician, it's always important to remember that there are already treatable conditions that mimic the symptoms of hereditary spastic paraplegia and cerebral palsy. So this is all great. We learned a lot, but what is the next step? The next step is really to think about how we're using this as a platform for clinical trials. And I would argue that similar to CMT, HSP is a great place to start because while you have 90 different conditions, 90 different monogenic diseases, and all different mechanisms of genetic diseases represented, everything converges on one central phenotype. So there's an opportunity for platform trials. So we're looking forward to being at a stage where the CMT field is now, where we have industry partners can conduct clinical trials in an efficient way and explore all of these different molecular mechanisms where appropriate. The reality for now the reality of rare disease drug development at the moment looks a little bit like this. There's a couple of challenges that I wanted to point out. I think many of them were already pointed out, but I'll just say that this is a long and difficult road, and there's many, many things that have to fall in place. There's a real need to build an infrastructure that can support both preclinical and clinical development. I'm highlighting here the different challenges at different stages of development, but in very simple terms, we go from understanding the clinical disease, which for many forms of HSP is where we're at, to understanding the molecular mechanism, to finding the right therapeutic entity, to proof of concept preclinical studies and appropriate models, to a pre-IND or to an IND-enabling preclinical package, and then hopefully on to phase 1, phase 2 clinical trials. The challenge is what comes after the phase 1, phase 2 clinical trial. How do we really translate this into pivotal studies and ultimately into approved drugs? So I'll share an example of one disease that we've worked on for the last 7 years, where we've gone a part of this way. And this is a rare group of hereditary spastic paraplegia related to a protein complex called the adaptive protein complex 4. You see the cartoon here. It's a protein complex with 4 different subunits encoded by 4 different genes on 4 different chromosomes. But loss of function mutations in any of these 4 subunits leads to the same problem. The protein complex can't form, and hence these 4 different genetic diseases share the same molecular pathology, but they also share the same phenotype and natural history. And just to illustrate that, I'm showing you videos of my first patients, and you see that they are about the same age. They have different mutations, including in different subunits, and they present very similarly. These children present usually in the first year of life with delayed motor milestones, with low muscle tone, and eventually progress to having spasticity, stiffness of their muscles, first in the lower extremities, and then in many cases, the rest of the body. Just to illustrate this was my first patient that inspired all of this work. I've met her when she was 3.5 years old. As you can see, she can walk at this time, although with some difficulties. This progresses very quickly to her needing more support of walking, a walker in the first instance. And you see her walking at 9 years. She's 11 years now, and she requires a wheelchair for ambulation. So this is a progressive disease that starts very early on in childhood. The defining feature of this disease, as the name implies, is spasticity. And I think maybe this is a paradigm for many rare forms of CMT and HSP. When we started working on this, we didn't know any of this. So the first step was to create a cross-sectional study. We looked at 150 patients to understand the spectrum of symptoms, and then to narrow down into longitudinal studies. So to illustrate or go over a lot of work that we've done since then, we've come to understand, I think, that like many diseases, and this has relevance to CMT, I think, as well, these genetic diseases, they're not just progressive degenerative diseases, but they in many cases also have a neurodevelopmental component, especially the childhood onset forms. These genes are important in brain development, and we start to appreciate or have started to appreciate that for AP4-related HSP. This is very important because it has implications for the therapeutic window and the expectations for therapies at certain stages of the disease. We've also learned that the earliest symptoms of this disease we can actually pick up before birth. Many children have changes on prenatal ultrasound and MRIs that we can at least use to refer or recommend genetic testing. On the basic science side of things, we had to do a lot of work. When we started, we knew that it was a recessive disease. We knew what the genes were, but there was very little known about the downstream pathways of this disease. And I'm summarizing the work of many people here over the last couple of years in one slide to say that understanding what is immediately downstream of the adaptive protein complex 4 enabled all the translational work that you'll see in the next couple of slides. So just to say, to the point that was raised during the first talk, it is really important that we understand the basic biology, at least to the degree that we have assays that we can use for developing therapies. So what I told you is that this is a recessive disease and a loss of function mechanism. So gene therapy to us seemed like gene replacement therapy to us seemed the most proximal way of approaching this disease when we started this about 7 years ago. And again, I'm summarizing a lot of work to say that for 2 of the 4 diseases, we worked through all the stages of developing an understanding of the biology, developing mouse models and cell-based assays. And then teams at UT Southwestern and at the University of Sheffield went on to conduct studies to develop and characterize an AAV9-based gene replacement vector for the treatment of SPG50 and SPG47, 2 of the 4 subtypes. And both have made it into clinical stages. For SPG50, the team at UT Southwestern, and you'll hear from Terry Pervolakis and Elpida Therapeutics this afternoon, have made it to a phase 1, phase 2 clinical trial that is ongoing at the University of Texas Southwestern. And there are plans and an improved protocol for a phase 3 study. And that's very unique for a rare disease. And I'll come back to the unique features that made this possible at the end. Following a similar pathway in learning from the first experiences with SPG50, we have now an approved IND and are hoping to start a phase 1, phase 2 clinical trial for the second condition, SPG47, using a different vector and a different route of administration here at Boston Children's later this year. Clinical experience with the first treated patient treated at the Hospital for Sick Children in Toronto has been published. This is a paper from last year illustrating, and I think the main accomplishment here was the speed at which this was developed. This went from a concept on paper to a first patient treated in 3.5 years. And I'm sure this afternoon we'll hear more about sort of what made this possible, but it basically took a lot of people working, and there's a lot of philanthropic support and taking a lot of risk also. So where do we go from here? This is a rare disease, and we struggle with all the challenges of rare diseases. Our natural history, our program has seen just over 300 patients with AP4-related HSP. The most common 3 types are listed here. And we're gearing up to do these clinical trials, both for SPG50 and SPG47. But how do we think about designing these trials? And in many ways, the phase 1, phase 2 clinical trials were approached from a very practical standpoint. We knew about the experience of an AAV9-based gene therapy delivered intrathecally for the treatment of giant axonal neuropathy, or GAN, which is a different rare disease. So we modeled a lot of our design after this. But the mission of the phase 1, phase 2 clinical trial for us really is to do 2 things. 1, to establish safety, of course, and 2, to see early efficacy signals. And in many ways, the latter point was informed by our natural history study. So this study that we're starting at Boston Children's Hospital will treat between 5 and 8 children with this experimental gene therapy. You see that we chose an age group between 1 and 5, and that's because of the natural history of this disease. We know that if you treat earlier, you have more, you stand to benefit more. That is, I think, very clear from all the natural history data that we have. We will deliver the gene therapy vector through an intracisterna magna infusion, hoping that this will improve biodistribution to central and peripheral, and spinal cord targets. The endpoints are listed here. Again, the primary endpoint in these studies is always safety, but you're really looking for an efficacy signal. And I'm listing with the asterisks, I'm listing all the endpoints that we've studied in our natural history study for the last 5 years, so they directly informed how we designed this. So we're hoping to pick up efficacy signal by having robust endpoints that mirror what we have in the natural history study. And in many cases, the patients that will be enrolled in this treatment trial will have had a lead-in phase coming from the natural history study. I'll briefly talk about the phase 3 trial design because that was an interesting experience with regulators, and I'm sure we'll talk more about this afternoon, but there's a unique opportunity here. As I've mentioned, this is a group of 4 different genetic conditions that share the same natural history. So we were able to design a phase 3 trial that doesn't require a placebo control group, but takes advantage of the fact that you can use matched untreated natural history controls of the 3 other subtypes as a control group. That's a very unique circumstance, and I think will hopefully allow us to, in a very rigorous way, test the efficacy of these therapies. And the regulatory agencies agreed with this approach. So there's some key considerations that I think are maybe in some ways specific to this rare form of HSP, but maybe are some food for thought for CMT. The first is, this is a rare but underdiagnosed condition. I'm very certain of that, not just because of my own anecdotal experience, but also because of the data on cerebral palsy. There's a clear mechanistic rationale. We know this is a loss of function disease. Gene replacement is the optimal treatment. We are lucky in the sense that this is a protein complex with 4 obligate subunits. So this protects us against overexpression-induced toxicity. If you overexpress one subunit, you don't get more complex function because the stoichiometry of the 3 other subunits limits how much protein complex is formed and stabilized. We take advantage of the fact that 4 different diseases have the same natural history. And the natural history study, I think to the point it was beautifully raised in the first talk, is the backbone for designing these clinical trials. I think this cannot be stressed more. I think natural history studies, robust natural history studies, are key for any rare disease. And then we have some emerging clinical experience. There's safety and tolerability data emerging from the first clinical trial that I think will inform how we do clinical trials for the other subtypes. So in summary, I hope I was able to show you that childhood onset HSP, like CMT, is a paradigm for the many challenges in rare diseases. AP4-related HSP is underdiagnosed, but there's a clear mechanistic rationale for gene replacement. Preclinical models and protein complex biology support a safe and targeted approach. Clinical trial readiness programs are key for really designing meaningful clinical trials. Phase 1, phase 2 trials should prioritize safety, but also looking for signals of efficacy. And lastly, pivotal trials remain a particular challenge in ultra-rare diseases. And I don't have any answers to solve this for all rare diseases, but I think as a general notion is I think we need more trials to gain experience, and I think that will propel us forward. So with that, I'm at the end. Thank you very much for having me for this short talk, and I have to thank the many people involved and many collaborators, our team and funding agencies. So thank you.
We have a question in the back.
Hi, Darius. Wonderful talk and good to see all the progress. I think in one of the slides, at least for the trial, I think I saw exploratory biomarkers, multiple biomarkers.
Yeah.
Can you expand on that? What are you looking at? And the second part of the question is, are you using any DHTs for any of this?
Could you repeat the second part, please?
Are you using any DHTs?
DHTs.
Digital health variables or
Yes.
sensors?
Yeah, thank you for the question. So I think in many ways in HSP, we're a little bit behind CMT. We're looking at blood-based biomarkers using very similar approaches that were talked about during the first talk. I think a blood-based surrogate marker would be tremendous for the field. So we're looking at the common markers identified in plasma serum, NFL, Olink panels, unbiased proteomics. I think all of that I think it's important work, and we're fishing really for the molecules that can tell us about disease progression in a shorter period of time. I think there's also a real need to understand what preclinical models can suggest about biomarkers. We know that all the HSPs convert to one central mechanism, which is axonal dysfunction and degeneration. So can we use preclinical approaches that screen using omic technologies for markers of axonal dysfunction and degeneration to identify those proteins or lipids or other targets that we can then in a more targeted way test in biosamples of patients? I think that's an approach that has a lot of merit. About wearable sensors, we are looking to test that as part of the consortium, with the idea being that in many trials, especially for gene-based therapies, with small cohorts, the patients will serve as their own control. So having a way of monitoring motor function in a decentral way, away from the clinic, at home in a relevant scenario, is really important. So I have high hopes for that. I think for small trials, that is a necessity. I think we need to explore that. Okay, thank you.